I'd like to thank the RNA Society for the Early Career Award. This is a huge honor for me. I'm a big fan of RNA, and while I'm a computational biologist by training, and my lab does a lot of molecular biology, chromatin biology, stem cell biology, and so on, we're primarily RNA biologists. And I'd like to thank my many collaborators over the years, my mentors, and particularly the different people who have passed through my lab at the Weizmann Institute over the past 6.5 years, my students, my research assistants, who made all of this possible. My lab is primarily interested in long non-coding RNAs. We're interested in understanding what kind of activities can a long RNA carry out in cells, how these activities are encoded in their sequences, and how they depend on different parts of their production. Understanding this is important for interpreting human genome sequences, as well as realizing the potential of RNA in the clinic. A particular sub-question we're interested in is how do these RNAs reach different positions within the cell? We know that lincRNAs display a variety of different localization patterns. This is from seminal work by John Rin and Arjun Raj a few years ago, where we can see that some lincRNAs are as cytoplasmic as an mRNA, whereas others are really enriched in a nucleus, and there is a really wide variety of different distribution patterns. If we look at mRNAs, we also know that most mRNAs are about 5-fold higher in the cytoplasm compared to the nucleus, but then there is a lot of diversity, and some RNAs are selectively exported in specific conditions to facilitate translation and protein production only upon specific cues. But what drives these processes remains quite poorly understood. If we look in the textbook, there is a quite well-defined pathways of different proteins being loaded on nascent RNA, then recruiting other proteins that are helping to push the RNA through the nuclear pore. And these have been implicated through seminal studies using either oligo-DT probes in the context of fish, or using vectors or mini-genes, and a lot of really beautiful studies in model organisms, which has taught us a lot about how these different transitions are happening. But in many cases, they were limited to looking at relatively particular types of genes and not necessarily at the average typical human gene, which would have about 8 exons and would have a relatively long transcription locus. So where does this selectivity come from is part where we know parts of the answer, but it remains largely unknown. And this is something that we've been interested in developing and using high-throughput approaches for understanding. One approach which we're using, a so-called massively parallel assay, or like John Rin likes to call these massively parallel RNA assays, are cases where we can take large numbers of different fragments which we can synthesize. So these will be thousands of oligos, each about 100 to 150 bases long, which can be introduced into vectors which have a particular typical localization pattern. For example, in this first work, we used a GFP, which is relatively efficiently exported from a nucleus. These thousands of plasmids are then transfected into cells. The cells are fractionated, RNA is extracted, and sequencing is used to quantify for each of these tiles how effectively it can affect the localization of the vector. So in the first work, we focused on RNAs which are efficiently exported from the nucleus and fragments of nuclearly encoded lincRNAs. And we identified regions which are shared between different lincRNAs derived from allotransposable elements in a particular orientation. This is a particular part of the allorepeat, which we call the serine elements, which we found was sufficient for driving about a 2-fold nuclear enrichment of the host RNA. We also found these sequences are bound by HNRPK, an abundant nuclear RNA binding protein, and in some subsequent work I won't have really time to dive into, we found that HNRPK needs to bind a particular place within these serine elements, and it recruits additional factors, including the U1 SNRP, for driving this nuclear enrichment. Another approach, and this is what I'll focus for most of my talk, is one where we can take and perturb different nuclear export factors and measure transcriptome-wide the effects of localization. This is work of Benjamin Zuckerman, a PhD student in the lab, which is about to come out soon. Here we used MCF7 cells, and we used RNAi to knock down either canonical members of the export pathway. These are proteins that are thought to be required for export of the vast majority of different long protein-related transcripts, as well as proteins for which it was suggested that they specifically affect export of partially processed transcripts. For each of these, we're knocking down the proteins and then extracting RNA and sequencing nuclear cytoplasmic and whole cell extract RNA, where the whole cell extract is used to obtain absolute quantification of nuclear cytoplasmic levels. And the first thing and the main surprising thing we found is that knockdown of NXF1 and knockdown of TREX components didn't really affect the localization of all the genes, but rather of specific gene subsets. When we compare these subsets, we saw that NXF1 and TREX in particular affected different groups of genes, whereas TPR knockdown, this is a factor which was thought to be somewhat specific, had a global and quite similar effect on localization of all genes, whereas U2AF2 knockdown had a relatively mild effect comparatively. So we focused here on NXF1 and TREX from now on. We also looked whether these factors preferentially affected link RNAs or mRNAs, and interestingly for NXF1, we found that knockdown of NXF1 preferentially caused nuclear enrichment of link RNAs over mRNAs. So this got us thinking about what's the difference between link RNAs and mRNAs. So one difference is that link RNAs typically have a small number of exons. And indeed, we saw that NXF1 preferentially affected expression of transcripts which had a single exon or few exons in general. And under these conditions, transcripts with many exons appear to be not affected at all. If we looked at other features, we also saw that longer transcripts, whether single exon or multi-exon, were also preferentially more reliant on NXF1 for their export. In fact, if we take mRNAs and match them to link RNAs based on different features, matching on the number of exon eliminates the difference between link RNAs and mRNAs, suggesting that what explains why link RNAs are more dependent on NXF1 is primarily the fewer exons that link RNAs have, typically 2 or 3, compared to mRNAs, which typically have 7 or 8 exons. Another feature which emerged was sequence composition. We found that TREX depletion preferentially affected localization of GC-rich transcripts, whereas NXF1 depletion affected more transcripts which were more A-rich, and particularly when they had many exons. We think this might be related to deposition of exon junction complex, which appears to preferentially interface with a TREX-dependent export and not as much with an NXF1-dependent export. This was all done by sequencing. We also wanted to see whether these trends actually hold up by imaging. So we use single molecule FISH. In these conditions, if we use a probe for oligo-DT probe for poly-A, we see that knockdown of NXF1, as has been seen in previous studies, causes a dramatic reduction in the signal in cytoplasm. But as we can see from the RNA-seq data, this reduction is driven by some transcripts and not others. For example, NORAD, a single exon cytoplasmic RNA in regular conditions, becomes strongly enriched in the nucleus when we knock down NXF1. We can quantify this. They see the same thing for other RNAs, which are single 1 or 2 exons and long. In contrast, in the same conditions, if we look at multi-exon RNAs, their localization is not affected at all. We can also use this approach to look at individual cells, and in cells we can combine staining for NXF1 to see how effective the knockdown of NXF1 was, along with looking at the localization of NORAD, and we can see that more efficient knockdown of NXF1 results in more drastic enrichment of NORAD in the nucleus, suggesting that there is really a causal relationship here. We then were interested to see which kind of sequences or structures are driving this NXF1 dependent exon export. So again, turn to these massively parallel reporter assays. Here we now use a backbone of beta globin without introns, which we got from John Stites. And now we can introduce into these backbones fragments of RNAs derived from 11 different genes, mostly mRNAs, which are long, single exon, and relatively efficiently exported. We then take these fragments, introduce these plasmids into cells, which either have a control siRNA knockdown or siRNA for NXF1, and then fractionate and sequence to obtain for each of these tiles to what extent it can drive export, which is dependent on NXF1. And indeed, in each of the genes we studied with this approach, we could identify at least one region, typically multiple regions, with this behavior. So for example, here we have a tile. This tile is driving export of the beta globin into the cytoplasm, but this activity is eliminated when we knock down NXF1. When we look at all of these tiles together, they have several interesting characteristic features. Look at them individually, we saw many of them appear to fold into predicted stable structures. And we see that in general, these tiles tend to appear closer to the 5' end of the RNA. They tend to be more GC-rich and therefore are predicted to be more structured than other tiles. They are more conserved in evolution, suggesting that this is functionally important. And if we look at CLIP data for binding of NXF1, we see that these tiles are binding NXF1 more effectively than other control tiles. So overall, this NXF1 dependent export appears to be driven by particular types of sequences which are found within these long RNAs and which are sometimes redundant but are sufficient for driving export. Interestingly, if we look at these features, they closely resemble the features which have been described for different viral elements which were shown over the past few decades to drive export of viral RNA, which also in many cases is going to be relatively long and unspliced RNA. And similarly, like these viral RNAs, which do not have any particular sequence or structure in common between them, we also see so far between the endogenous sequences, there is this shared features of being GC-rich and being structured, but we don't yet see any particular shared motif. We could then take these styles and cross them with data from the ENCODE project, ECLIP data on binding of RNA binding proteins, and then ask which RNA binding proteins preferentially bind within regions which are mediating export. And one factor which emerged from this analysis is RBM15, which has been previously interestingly implicated in an XF1-dependent export, and also has been implicated in M6A modifications. It's been kind of an optional component of the M6A writer complex. So what we try to do next is to use the same reporter approach, combining it with knockdown of RBM15 or knockdown of WTAP, which is a core component of the M6A writing complex. Indeed, if we look at fragments which are driving an XF1-dependent export, these tend to be enriched with M6A modifications. When we knock down RBM15, we actually don't see any effect on export. So RBM15 binding by itself does not appear to be crucial for export, but if we knock down WTAP, we see that the ability of these fragments enriched with M6A modifications to drive an XF1-dependent export is abolished, suggesting that on top of these specific structural elements, M6A modifications are also important for an XF1-dependent nuclear export. So overall, what we learned is that some transcript classes are preferentially dependent on XF1. These tend to be transcripts with one or few exons, longer exons, or many exons but AU-rich ones. Within these transcripts are specific structured M6A modified sequence patches which are mediating an XF1 export. And in contrast, if we look at dependence on TREX, it has other determinants. So these are preferentially GC-rich or particularly C-rich transcripts, and these tend to be transcripts whose baseline export levels are not as high as others. Why is this important? So first of all, if we think about link RNAs, new link RNAs arise in evolution all the time, and they would typically have few exons, and they would in many cases be relatively AU-rich, which would mean that their export would be relatively inefficient and very NXF1 dependent, unless they manage to recruit elements which facilitate particularly efficient export. Also, this means that different processes in the cell can use this specificity of different factors for regulating export of different gene subsets. And indeed, we see that genes that are known to be affected in their export by different viral proteins preferentially overlap with genes which are sensitive to NXF1. We can think, for example, of interferon mRNAs, which tend to have few exons, which are preferentially targeted by different viral proteins, and we see that these genes also tend to be preferentially targeted by NXF1 export pathway and not other pathways. With this, I'd like to end and thank members of my lab, and particularly Binyamin and Maya, which were involved in the work I talked about mostly, and mention that we're always looking for great postdocs, and to again thank the RNA Society for the Early Career Award.